Assalamu alaikum students. You know that multicellular organism is made up of many cells like human being, plants and other multicellular organisms. So cell cycle will take place in the cell only. Today we are going to study the stages of mitosis and cell division. In the cell there is a cell division based on the phases, that is G1, G2, M and S phase, where M and S phase are the main part of the cell division. An adult human contains an estimated 100 billion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cell, this process is accomplished by series of well-organized steps called mitosis. Every day, our body must produce millions of skin cells to replace the loss of normal activity. Each of these cells must have a complementary genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histogen proteins, forming nucleosomes. These beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and formation of sister chromatids is one part of the entire cell cycle to prepare for cell division. Cell goes through interphase, which can be divided into three distinct phases. During G1 or gap-on phase, all the organelles and cytoplasmic components, including the centrioles in animal cell, replicates. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap 2 phase, all the enzymes needed to aid in the process of cell division are produced. Most of the eukaryotic cells spend a great deal of time in interphase and a very short period of time actually during process called mitosis. Now here actually the process of mitosis will start, and the cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase and telophase. In prophase, the chromosome condensed and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton dissembles as the spindles begins to form. In animal cell, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelopes break down. Towards the end of prophase, chromosomes attached by proteins in their centromeres called kinetochores to microtubules form each pole. The chromosomes towards the equator of the cell during metaphase, all chromosomes are aligned at the equator of the cell metaphase. Metaphase begins by the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filament surrounding the inside of the cell circumferences. Chromosomes cluster at the opposite pole and begins condensing as the nuclear envelope reforms around them. Spindle apparatus dissembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. At this point, cell division completes in animal cell by forming two daughter cells that will be exactly like their parent. In plants, cell wall cannot be constricted by actin fibers. Vesicle from an expanded membrane form a partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the content of the cytoplasm between the two identical daughter cells. Exactly like their parents' cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately, and if it is not, the cell cycle will stop at the checkpoints and correct or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that chromosomes have attached to the spindle for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequency as well. In humans, our skin cells have high turnover rate due to wear and tear and go through mitosis very frequently. This process allows for growth and repair to prolong overall physiology as well as life itself. Hopefully now you might have understood that how mitosis takes place in living organism. So like this, chromosome will split up into two identical daughter cells. Thank you.